Hello friends, myself Mr. Sagar Tukaram Kadlak from Gokhale Education Society's Arts, Commerce and Science College, Jawhar. Today in this video we are going to study about our next topic, that is genetic drift. It is the topic from SYBSc Zoology paper first, semester 4, unit number 2. So genetic drift was explained by Swell Wright, or the Swell Wright explained the idea of a genetic drift in 1931. Therefore, this genetic drift is also called as, or also known as, Swell Wright's effect. It refers to change in a gene frequency brought about purely by a chance in a small population. In small population, some gene may increase or decrease their frequency by chance. So this lead to the changes in a gene frequency within the population. Thus, the gene pool of a population is changed without considering the usefulness of the particular traits in the population. Such a chance and these changes in the gene frequency take place due to the sampling error. So this chance of gene frequency variation is due to the sampling error. Now the smaller size of a sample will give me the chance of deviation from the perfect result. If we consider that the smaller size of a sample, it will give a greater chance of deviation or greater chance of variation with your result. That must be in a algorithmic form or a numerical form we will expected in the sample. So this disproportion of the result or the changes found in the population we can simply call as the sampling error. If a new generation draws its alleles at a random, then the larger the sample size, the better will be represent gene pool of the previous generation. So if we consider the idea about sampling error, a tossing experiment of a coin, when a coin is tossed for several times, the probability of occurrence of head and tails is more or less. However, when some coin is tossed only for 3 to 4 times, it is possible that the all will be head or a tail purely by a chance. Thus, it will give you a clear-cut idea. The smaller the sample size, it will greater chance of a deviation from the perfect result, as we see from the sample or the example by considering a coin toss. So the gene pool of that generation will be varies in a previous one. So this chance of fluctuation in a small population goes on continuously until the one allele or one trait and its partner trait is lost and the another one is fixed. That is, there will be a fixation of the particular dominant one or the recessive one, but the elimination of heterozygous condition, as well as we can simply explain as a one dominant will be remain and the recessive will be eliminated, or the recessive will be continued in that population and the dominant will be eliminated. So this is the general explanation regarding the elimination of a gene from the particular population. So thus the allelic frequency in a small population allowing drifting by chance event from generation to generation. So this force of chance is called as the genetic drift. So chance of fluctuation in a small population goes continuously. So this process may yield 100% frequency of one allele by losing the another or other trait in the population. That is, we can simply consider as P is equal to 1 for the one allele and pair Q is equal to 0 or vice versa. So this is a very clear that a random fluctuation in the population may lead to a complete fixation of one allele or the another. An allele can be completely lost from the population by a chance. So this will be known as a Sewall-Wright's effect or the chance of sampling error or the genetic drift. Now this genetic drift may be induced by a bottleneck effect. So now what is meant by a bottleneck effect? This term was coined by a Stabin, and according to him a large population sometime a relatively few individuals survive out of them from the progenitor or the parents for the population in the next season. This seasonal or a cyclic or yearly phenomenon is known as the bottleneck effect, which established in the population when it contracts to the significantly smaller size over a short period due to some random environmental events. So there will be a certain or few individuals remain survive because of the environmental events such as a flooding or excessive raining or the earthquake. From a large population a few only survive, and this reduction in that number of individuals from the population we can call it as the bottleneck effect. This seasonal or cyclic phenomenon is called as the bottleneck effect. Now this bottleneck effect we can simply consider with the example when a population of lemmings, or we can simply call as the small rodents, lemmings, living in a Scandinavian mountain reaches an extraordinarily high in every 4 years. They migrate to the coast, jump into the sea and dies. This population gets re-established by a few lemmings remains in the mountain. So those are able to survive at the mountain level. They will continue for their race, or they will maintain their race within the population, or they can continue their trait as a in a next generation. Now this changes called as the bottleneck effect. Only few individuals will be remain in that place or in that population. Now founder effect, these are the another one term coined by the idea of founder effect, the Ernst Mayr in 1942. This term coined by Ernst Mayr with respect to a newly established population. So this term is mostly used or with the respect of newly formed or established population. According to him, when a new but isolated population is established from the parent population, its gene pool differs from that of the parent population due to the sampling error. It's leading to the genotype as well as phenotype differences in them. So these differences from parent to the next new generation is due to the sampling error Okay, now these various evolutionary forces acting independently in the two population further increases these differences in the gene pool. Such differences lead to a genetic divergence. Now we can consider that the evolutionary changes induced by the founder effect in the both population can cause the two kind of changes. The first one is that in comparison to the large population, the descendant small isolated population possesses an unusual character, possesses a different one characters or unusual characters which not found in the previous one. The second one is that the large population descent from the new migrants may differ from population from which the immigrants come. So both kind of the population may tend or may contain to different genetic traits or alleles in that population. So such a differences or such a changes in a gene pool called as the founder's effect. It is mostly due to the sampling error. Okay, so this is all about genetic drift, bottleneck effect and founder effect in this video. Thank you.